Good day, everyone. I am Ayabuni, and our topic for today is seminal fluid. Our objectives at the end of the lesson, the students will be able to first discuss the compositions of sperm, explain the procedures for the collection and handling semen specimens, and lastly, describe the normal appearance of semen and three abnormalities in appearance. For our topic outline, introduction, semen composition, spermatogenesis, specimen collection, macroscopic examination, and lastly, sperm morphology. So let's start. Sperm cells are male reproductive cells that originate in the testicles. Semen is composed of four fractions that are contributed by the testes, epididymis, seminal vesicles, prostate gland, and bulbourethral glands. Each fraction differs in its composition, and the mixing of all four fractions during ejaculation is essential for the production of a normal semen specimen. The testes are paired glands in the scrotum that contains the seminiferous tubules for the secretion of sperm. The external location of the scrotum contributes to a lower scrotum temperature that is optimal for the sperm development. Germ cells for the production of spermatozoa are located in the epithelial cells of the seminiferous tubules. Specialized circulatory cells provide support and nutrients for the germ cells as they undergo mitosis and meiosis, which is called spermatogenesis. When spermatogenesis is complete, the immature sperm or non-motile enter the epididymis. In the epididymis, the sperm mature and develop flagella. The entire process takes approximately 90 days. The sperm remains stored in the epididymis until ejaculation, at which time they are propelled through the ductus deferens, or it is also called vas deferens, to the ejaculatory ducts. For semen composition, semen is composed of four fractions that are contributed by the testes, epididymis, seminal vesicles, and prostate glands. So we have four fraction. First, spermatozoa, which contain 5% of semen. Second is seminal fluid, which contains 60 to 70% semen. Third is prostate fluid, which contains 20% to 30% semen. And lastly, bulbourourethral glands contains 5% of semen. So what is spermatogenesis? So spermatogenesis, it is the complete process of cell development. Spermatogenesis is a process of developing male gametes known as sperm within the male reproductive organs, the testes. In this process, each sperm or haploid containing a single copy of each chromosome, in order to create the haploid gamete, a cell undergoes the process of meiosis, in which the genome is replicated and into divided twice to produce four haploid gametes. This process generally occurs in the seminiferous tubules of the testes following different stages. It is followed by maturation in the epididymis, where they are secreted in the form of semen along with the glandular secretions. Excuse me. This process begins during puberty ends only when the individual dies. The complete process of spermatogenesis in males are carried out by the actions of Leydig cells, hypothalamus, and pituitary gland. The quantity of these sperms gradually reduces with the age and finally leads to infertility. The process of spermatogenesis occurs to create mature male gametes, which then fertilize female gametes to create a zygote, a single-celled organism. This results in cell division and multiplication to create a fetus. For a healthy offspring, the number of chromosomes must be maintained properly across the body, as failure can lead to some abnormalities. So approximately 90 days for the duration of the sperm, according to Strassinger. And according to graphs, we have 74 days, which involve several phases. These are the spermatocytogenesis, meiosis, and spermiogenesis. So let's go on to specimen collection. So the preferred method of semen collection is by masturbation. This procedure ensures the opportunity to collect the entire ejaculate. Collection should be performed after 48 to 72 hour continence or abstinence from sexual activity. Specimen collection. Specimen collection containers should be clean glass or plastic and have wide opening. Specimens awaiting analysis should be kept at 37 degrees and extremes in temperature must be avoided. First portion of the ejaculate is missing. Decrease sperm count, specimen will not liquefy, and increase pH. For last portion of ejaculate is missing, the semen volume is decreased, the sperm count is increased, and the pH is decreased. And specimens will not club. For the sperm morphology, liquefaction. Once the specimen arrives in the laboratory, it is monitored for a period of liquefaction. If coagulation did not occur, it should be reported. Normal liquefaction between 30 and 60 minutes, and for the abnormal liquefaction, it would be more than 60 minutes. To obtain accurate sperm counts, specimens do not dissolve should be treated with amylase and bromelain for removal of mucus. For the appearance, semen is opaque and can exhibit several normal colors. Typical color include Typical colors include gray, white, and light yellow. The higher the flavin concentration of semen, the darker the yellow color may be. So, deep yellow color if associated with certain drugs, and brown or red colored which contain blood. A highly turbid semen specimen usually contains leukocyte and may indicate a reproductive tract infection or inflammation. For the viscosity, so viscosity may be assessed while measuring specimen volume or when pipetting the specimen for other tests. So slightly viscous and dispensed drop by drop would be a normal semen, and watery would be considered an abnormal semen. Increased viscosity is demonstrated by the formation of fluid of the specimen is dispensed from a pipette. Next is volume. That should be volume. So, semen volume is measured by using a serological pipette or small graduated cylinder. So, volume is recorded in milliliters to 1 decimal place. Normal semen volume would be 2 to 5 ml for complete ejaculate. For increased volume, it may be seen following periods or extended abstinence. For decreased volume, more frequently associated with infertility and may indicate improper functioning of one of the semen producing organs, primarily the seminal vesicles. For the sperm morphology, so this is the picture of a normal sperm. So head must be 5 micrometer long and 3 micrometer wide, according to Strassinger, 4 to 5 in length, and 2 to 3 meters in width, according to graphs. So it appears oval when viewed from the side and can be mistaken for an abnormal form by inexperienced observers. So the tail piece should be 45 micrometer long according to Strassinger and 50 to 55 meters in length according to graphs and varies in thickness from 1 to 0.01 micrometer neck to tip. For the neck place, attaches the head to the tail and the midpiece. For the midpiece, it should be 7.0 micrometer long and is the thickest part of the tail because it is surrounded by mitochondrial sheath that produces the energy required by the tail for maturity. So this is the pictures of or illustrations of an abnormal sperm. As you can see, we have double head, two, two tails. So, abnormal sperm morphology occurs as an anomaly of either the head or the tail piece or both. As you can see in the picture, identified abnormalities in head structure include double heads, giant and amorphous heads, pinheads, tapered heads, and constricted heads. Abnormal sperm tails are frequently doubled, coiled, bent, multiple tails, and variation in tail length. An abnormal long neck piece may cause the sperm head to bend backward and interfere with motility. So here's the reference. So that's all, everyone. Thank you for listening.